So good afternoon, everyone. Like she said, my name is Jada Harvey, and I was in the Pratt Hoover lab at the University of Rochester. And my project was on the spatial and neighborhood analysis of immune cell types in lung tissue. So to start with a just an introduction or a background, as you may know, the lungs are a very important part of the respiratory system, as they are responsible for the gas exchange between oxygen and carbon dioxide. So each of your two lungs are separated into lobes, your right lung into 3 lobes and your left lung into 2 lobes to leave room for your heart. So the lungs consist of the bronchi that split off from your trachea into smaller branches of bronchioles and further into the alveoli. So when you inhale oxygen, it moves through all of these components to be supplied to the bloodstream, and carbon dioxide from deoxygenated blood moves back through these components and out when you exhale. So what is the objective of this project is to define and classify cellular neighborhoods and observe localization and structure of immune cell types. So to start off, our first step was fluorescent staining done through our phenocycler fusion. So this whole process is just to ensure that the phenocycler run goes successfully. So we start with our pre-antibody staining process. So this ensures that our tissue is adhering to the slide. Our slides are prepared in formalin, so getting some of that formaldehyde off of the slide and keeping our tissue from drying out, as well as our last step with the antigen retrieval, which is increasing our ability for our tissue to bind with the antibodies. So our antibody staining is where we receive our barcoded antibodies from Akoya Biosciences, and we simultaneously stain our tissue with all of our antibodies of interest. So next is our post-antibody staining process, where we complete our fixation in PFA and methanol, and we mount our flow cell, which is used to run things over the slide without the possibility of damaging the tissue. So lastly is our phenocycler fusion run. So our phenocycler is an automated system that allows or that works to bind our tissue that has been stained with our barcoded antibodies to our phenocycler reporters. So our phenocycler reporters have these oligonucleotides that are complementary to the barcoded antibodies, and they contain the fluorescent dye. So in our phenocycler works in cycles. So each of these cycles contain our DAPI, which is our nucleus marker, and our three antibodies and 3 other antibodies. So we work currently with our 27 antibody panel. This is an abbreviated list, but is focusing on the main cell groups that we look at. So next is the start of our analysis in our QPath software. So in our QPath software, we're able to view our fluorescent image as well as phenotype our various cell types within the tissue. So the first image here is just our fluorescent image. So you're able to manipulate colors as well as make annotations to focus on specific sections of your cell or the whole slide image. So in this step, you also do your cell segmentation, which is segmenting individual cells and through the nucleus, and QPath generating a boundary around these nuclei. So right now we use our Stardust scripts to do our cell segmentation, but we've lately been working on new ways to improve the cell segmentation. So next is our classification or our machine learning process or a portion of the QPath analysis. So in this, it's just teaching our software what we want to identify as each cell type or each of our cell types of interest. So in this example, we are using our object classifier, which uses points to identify each cell type. So for example, we have our CD20 and our CD4 cells. So the points for the CD20 are identifying the cells that we want the system to identify as CD20, and these points are identifying the CD4 cells. So once we have identified as many points as you would like or that you think you need, you will run your training, and it should look something similar to this. So the trainings are based on the criteria of the points that you have identified. So using the criteria of the points, it highlights other points, or it highlights other cells that fit into that criteria. So also with the training, you're able to correct any misidentifications. So I think that's a good aspect of QPath as well, but it could get a little harder when you're dealing with cells of varying intensities. So if you have a cell type with signal or a different intensity in two groups of cells, but you need both intensities to be identified as one cell type, that can be kind of difficult. So this last image is just an example of what, so when you complete your classifications in QPath, that is the data that you are exporting into CytoMap that I'll talk about on the next slide. But these classifications are then exported to CytoMap and used for the CytoMap analysis, and then exported back into QPath. So I just wanted to include an example of what that would look like. So now for the CytoMap analysis. So CytoMap is another analysis tool that we use for the classification of cellular neighborhoods. So as I stated before, you use the classifications that you generate in your QPath to export into CytoMap. And CytoMap generates cellular neighborhoods based off of your classifications and your trainings of various cell types. So CytoMap generates these cellular neighborhoods based on the abundance of cell types within each neighborhood. So neighborhoods are represented in CytoMap by regions. So each region is identified by color. So I like to just call this a region map. And CytoMap also equips you with a chart, so you can identify what cell types or what phenotypes are most representative in each of these regions. And also through CytoMap, you are able to perform your dimensionality reduction, which is your UMAPs, as well as your heat maps, and they're all based on your initial cellular neighborhood analysis. And as I stated before, the cellular neighborhood analysis is able to be exported back into QPath and laid over your original fluorescent image. So a big part of working with the CytoMap and QPath was a lot of experimentation, just trying to figure out what works best. And the biggest realization that we were able to come to was in CytoMap that working with smaller sections of cells were what worked best in being able to define specific cellular neighborhoods. So this is a QPath image of a whole slide, and this is the CytoMap image of this whole slide. So as you can see, when like our project is focused on looking at specific immune cell neighborhoods, but with this whole like array of colors, it's kind of hard to focus in on one type of cell or one type of neighborhood of cells when you have this vast scale of cells everywhere on the slide. So also this is an example of a UMAP created through the whole slide image. So here we have an example of a secondary lymphoid structure that we found in one of our tissues. So this is also something that helped us realize that working with smaller sections was best, and it identified more essential information that we were looking for, and why we started to focus more on immune cells. So here we have the CytoMap image and the QPath image, and you can see that it kind of reflects exactly what is seen in the QPath or the fluorescent image. We were able to identify clear CD20 B cell zones, our CD4 T helper zones, and as well as our the yellow and blue zones. So they both have the subsets of T cells within them, but they're identified as different regions because of the distribution of these two subtypes. So next we have an example of an airway-associated lymphocyte cluster. And the first thing that we noticed about this, this lymphocyte cluster was the organization. Again, just like the secondary lymphoid structure, we were able to define clear zones in this cluster. So represented in the yellow and the green are our T cells. So we have a CD4 zone and a CD8 zone, as well as for the B cell zone. It's represented through the burgundy and the red. And because this is airway associated, we use our pancytokeratin, which is our epithelial marker, to identify that this is associated with an airway. And just as before, you can clearly see even in QPath that these zones are clearly defined. So next is our vessel-associated lymphocyte cluster. So we noticed a very big difference between the vessel-associated clusters and the airway-associated clusters. Again, for the airway-associated clusters, we noticed there was a certain kind of organization that just wasn't here in the vessel-associated lymphocyte clusters, as well as the localization of the B cells. These vessel-associated clusters tend to be a lot more T cell oriented, and just the population of CD20, which is our B cell markers, are just not very not very populated. So, and also these vessel-associated clusters, they tend to be very few in number. That's why I decided to analyze them both together, just so you could kind of see. It's kind of harder when you're it's kind of harder to analyze with only about 100 cells. But as you can see, like I said, in the cytomap analysis, it tends to be more of a mixed type of regions where there are different distribution of these lymphocyte markers. So it has the CD20, it has the CD4 and the CD20, as well as the CD4 and the CD20 here, but the distribution is different, therefore defining them as different regions. So lastly, with the summary and conclusions, so we were able to successfully define regional localizations of immune cell types in vascular and airway associated clusters. So we were able to identify that in vascular associated clusters, it tends to be more T cell centered or T cell oriented, where B cells are just not very, they're just not very populated in these clusters, as opposed to the airway associated clusters, where there are clear zones or clear defined neighborhoods of T cells and B cells. And our second point was that there are distinct differences in organizations of airways versus vascular associated lymphocyte clusters. So the airway associated clusters were very organized, again, as the B cell and T cell zones, whereas the vascular associated lymphocyte clusters were very mixed and very intertwined with the B cell and T cell clusters. So the airway associated lymphocyte clusters, through a little reading, have been described as inducible bronchus associated lymphoid tissue. So this can be as a result of exposure to allergens over a period of time. These clusters can form, and the localization of the vascular associated clusters may be indicative of some type of inflammation. That's why they are more T cell oriented. So I'd just like to acknowledge Dr. Gloria Pry-Huber, the principal investigator in my lab, as well as Dr. Jeffrey Perkerson. I'd like to thank all of the other people in the Pry-Huber lab. I had an amazing summer with them. I learned a lot, and I'd also like to thank the NIH HuBMAP program for giving me this opportunity. Maya and Dr. AJ, they were very instrumental in getting all of this together and ensuring that we had everything that we needed. So, thank you.